Okay, welcome back. So this is the last video for this week where we're talking about reading scientific papers, and what I wanted to do in this video is just give you a little bit of background to help you understand the paper that you're going to be reading that you'll then be writing the missing abstract for. When I was reading this paper, I was trying to read through it from your perspective, and there were a few things that I think might be confusing or things that you haven't learned about before, so I wanted to take some time in this video to clarify those things so you can focus on the paper itself and not get caught up on some of the methods, for instance. So in this video, I'll talk about this gut microbiota paper that you'll be reading for your intro. They'll be reading to write the abstract for. So one of the things that they mention in this paper is that they use the hypervariable region of 16S in order to perform their study. And so if you haven't heard about this region before, this is a commonly used gene to identify prokaryotes in particular, so like bacteria, for instance. And if you took BIO2 here, think back to BIO2, we actually talked about this and worked with this gene in BIO2 lab. If you didn't take BIO2 here, that's totally fine. I'll go ahead and give you all the information that you need to understand what's going on. If we think back to some of our kind of basic biology where we're thinking about translation, where we're taking information contained in mRNA and converting that to the form of amino acids and proteins, that process is performed at a ribosome, which is shown here in the green. And just for some context, this 16S rRNA that we're looking at, this is a component of the small ribosomal subunit. So that's where this particular gene functions and is located. And this slide looks busy, but I'll walk us through it. So here on the left, this is what the actual folded structure of that 16S rRNA looks like. And there are a couple different regions within this gene that make it a good gene to use for the types of studies that the authors of this paper did, for instance. So if you read the paper, you'll notice that they say they used PCR primers to amplify the 16S rRNA region. And so if you look down here, and if you think about, if you've learned about PCR or done PCR before, remember we have these two primers that bind to these particular regions. They come in opposite directions and then they amplify everything that's in the middle there between those two primers. And so for this 16S rRNA gene, they use primers specific for this particular region to amplify the 16S rRNA, which is what they examined in their study. And so if we look here in particular, the region of this folded 16S rRNA that they have the primers binding to is called this conserved region, which is the stem. If it's conserved, which means it's the same across many different bacterial species, that means you're going to be able to use the same PCR primers because it's the same sequence to be able to bind and match to this same portion of the 16S rRNA across many different types of bacteria. The other part of this gene that I want to call our attention to is called this variable region. So here, this hypervariable region, this is the part that actually varies between bacteria. And so when we're doing analysis, what we're doing is we can look at the actual nucleotide sequences, the nucleotides of this 16S rRNA region in this variable region, and that'll help us to actually identify what types of bacteria are present in our sample. And in this particular study, they mentioned they use something called high throughput sequencing. And so what that means is, again, if you think back to BIO2 lab, if you took it here, we were isolating single bacterial colonies from a plate and just examining the 16S rRNA of that particular bacterial colony. High throughput sequencing doesn't do that. What it does instead is you can take a sample from the gut, for instance. In this paper, they obtained their samples from feces. And here in this picture on the right, each of these different colored broad-looking things here is a different type of bacteria. And so in high throughput sequencing, what you can do is take a sample and then examine all the 16S rRNA in the gut. So not just from a single colony, but you can see overall how many different 16S rRNA genes and how many different types of bacteria are present in your whole sample. And so that's why this is called high throughput sequencing, because you're processing lots and lots of data at once, not just from a single species. So what you can do is take your sample and then run it on what's called a sequencer. So you may see in the paper they mentioned something called a 454. That's just a type of sequencing technology that allows you to do this high throughput, where you're looking at 16S rRNA from a bunch of different types of bacteria at the same time. So what you do, or what you get back after you run the sample in that sequencer, is a bunch of nucleotide sequences or base pair sequences. And remember, we said that that particular region of 16S varies across different bacterial types. So what you can do is then take those unknown sequences, compare them to a database of known sequences. You use a database called BLAST, or Basic Local Alignment Search Tool. Again, if you took BIO2 here, we used this in BIO2 activity for your genes and disease project, and we talked about it throughout the semester. So what you're doing then is taking those unknown 16S sequences, comparing them through BLAST to a database, and then finding a match. Hopefully I have some Kristen Wiig SNL fans watching this who will appreciate my Target Lady reference. It's a match. So once you find a match, then you can know that your unknown 16S sequence matches that known sequence in the database, and so you can then know what type of bacteria you have present in your sample. Another thing that's mentioned in the video that might be good to clarify is they also talk about, in addition to looking at what bacteria are present, they also looked at a particular type of macromolecule. Again, that's something, if you took BIO2 here or you've learned in a previous class, we talked about the different types of macromolecules. Fatty acids are one of them, and in particular, short-chain fatty acids, or SCFA, are something that's produced by certain bacteria, and so they talk a little bit in the paper about why that might be beneficial. But I just wanted to show you or explain a little bit what that is. So here we have some examples of short-chain fatty acids that they talk about in the paper: acetic acid, propionic acid, butyric acid. They're called short-chain because these carbon chains here are all relatively short. So that's what they mean when they say short-chain fatty acids. And again, just a little bit of methodology. They identified what type of fatty acids were in their sample using something called mass spectrometry. So you run your sample through another machine, shown here, and then what it gives you are what are called these different spectra, and there are different peaks depending on what type of fatty acid you have. So here's acetic acid, there's a peak down here, versus a different type of acid has a peak over here. So by obtaining these spectra for different fatty acids that are unknown at the time you run them in this machine, you can then obtain these patterns and then determine what type of fatty acids you have in your sample by matching these peaks. And then once you get to the methods or the analysis section of this paper, they talk about the particular statistical tests that they use to analyze their data. And so I wanted to take a minute to explain and refresh, because we have talked about this a little bit so far this semester. We've talked about when you use parametric versus non-parametric statistical tests. So we've made histograms in class. So down here at the bottom we have two different types of histograms, and histograms allow you to look at the distribution of your data. And so here on the left we have an example of a normal distribution. It's like a bell-shaped curve. And if you have a normal distribution, that means that you can run a parametric test. So when we talked about ANOVA a couple weeks ago, one of the requirements for being able to run an ANOVA is that you have a normal distribution of your data. If you have something on the right that looks like not a bell-shaped curve, but maybe has two different humps there in the distribution, a bimodal distribution, so bi meaning 1, 2 modes here, 2 peaks, If you have a distribution that's non-normal, that's when you need to use something called a non-parametric statistical test. So we're not really going to talk about these types of tests in BIO100, but just so you know the difference when you would use parametric versus non-parametric. And one type of non-parametric test that they actually use in this particular paper is called a Kruskal-Wallis test. And again, you don't need to know the details, but just so you understand, this is the non-parametric equivalent to an ANOVA. So if you had data that you were comparing 3 different groups, for instance, just like we talked about, that's when you would use an ANOVA. But if your data is non-normal and doesn't have that bell-shaped distribution, then you could use this Kruskal-Wallis test. And just another heads up, in this paper they use a few statistical tests we have already talked about this semester, so go ahead and keep an eye out for those, and hopefully you'll be able to remember that we've already talked about those types of tests. And then the last thing that I wanted to explain is that you'll see in one of the figures of this paper, they show something called a dendrogram, and they say that they constructed this dendrogram using something called hierarchical clustering. And so the key word here is clustering. So I have an example of a dendrogram here shown on the bottom. This is a dendrogram made from that MT-CARS data set that we've been talking about throughout this semester. And so again, we don't need to know the details of this, but I just want to show you how to interpret these types of figures. So in these dendrograms, each one of these branch points here, in this case, because we're talking about the MT-CARS data set, it's a different type of car. And so what the people who constructed this dendrogram have done is they've used all those characteristics within that MT-CARS data set, like MPG and horsepower and displacement. All those different characteristics are different for each of these different types of cars. And so what you can do is for each of these cars, so like for the Camaro Z28, for instance, you can say, okay, based on all of these different types of criteria, that particular car is most similar to this Duster 360 because it's closest here in this clustering, so they're clustered together, they're close branch tips. And so that means that based on all of these characteristics, MPG, horsepower, displacement, those two types of cars are very similar and clustered together on the dendrogram, whereas those two types of cars are very different from this car, the Merc 280C. I don't know what any of these types of cars are. I don't know anything about cars. Oh, Toyota, I have heard of that one. Okay, so the idea here is when you're reading these dendrograms, what you're looking for are tips that are clustered closest together. That means they're the most similar to each other, and things that are farthest apart are the most different. So here in this particular dendrogram, you see they have colored boxes. So this is a red box for one particular cluster, another box shows another cluster, and then a third cluster over here. And then just another plug, when you're reading this paper, you'll see that the authors say they use the R package H-Clus to make this the dendrogram they use in the paper. So given your R skills that we've gained so far, this is something you could actually do if you really wanted to, is use that H-Clus R package to make your own dendrogram. So I've taken some time to explain what I think are some of the more confusing parts of the paper, but that doesn't mean that you're not still going to have questions as you read the paper, and that's totally fine. So what I would recommend is taking notes and jotting down as you're reading what you don't know. Look that up later or ask other people. And like I said, that's totally fine. We don't expect you to understand everything that's in this paper, but as you're reading it and looking things up, that's how you're going to build knowledge and learn. And so if you read another paper later on that uses similar types of techniques to this one, you're going to be familiar with those now because you've taken the time at this point to learn more about those techniques. So if you have questions, that's great. That's totally fine. We'll do our best to answer them because we'll actually be talking about this paper in the next week where we analyze scientific papers as well. Okay, so now your assignment for this week is to write the missing abstract for this paper that we provided for you. So go ahead and read that paper. On Canvas, there's a quiz where you're going to fill in some questions that'll help you write that missing abstract. Then you'll write the full missing abstract draft, upload that on Canvas before you come to class, and then when you come to class, bring a hard copy and a digital copy of that abstract draft with you, and we'll do some peer review, we'll revise it, and then submit a final abstract in class. So go ahead and read that paper, and hopefully it'll be a little bit clearer for you after watching this video.